Thank you for coming to our session. I'm Siddiq from the University of Chicago. I work in the Thornton lab, and today I'll be talking to you about ADH adaptation in Drosophila. Basically, using ancestral sequence reconstruction to test a classic hypothesis of molecular adaptation. So, generally in evolutionary genetics, a large program is to understand how changes in genotype lead to changes in fitness. And for those of us who are interested in the biological processes and the molecular mechanisms by which this happens, we have to specify and test causal links that connect variation at these two levels. For protein adaptation, which is what I work on, this requires characterizing how changes in genotype lead to specific changes in molecular phenotypes, how that in turn leads to changes at the organismal level, and how that affects fitness given the organism's natural history and ecology. So this requires testing hypotheses across biological levels, and that is hard enough on its own when we're working with species or between populations that exist today, and it's particularly difficult when we're interested in hypotheses of historical divergence. For example, if there is an adaptive trait that evolved in one species, just like with populations, we would want to know what are the underlying genetic changes, how they function, and how they might affect fitness. But unlike with populations, the variants we need to compare to study that don't exist. We are making predictions about things that happened in history. As a result of this, studies of adaptive divergence have largely relied on correlational data, and the ADH system in Drosophila melanogaster is an iconic example of that. So to give a brief background, Drosophila melanogaster has uniquely high ethanol tolerance among species in its subgroup. Here you see that between Drosophila simulans and Drosophila melanogaster, strains of melanogaster can tolerate much higher levels of ethanol than can strains of Drosophila simulans. ADH is the protein that catalyzes the first step of the ethanol catabolism pathway. And in comparisons of ADH activity between Drosophila melanogaster and other species, we see that ADH is more active. Furthermore, the first ever McDonald-Kreitman test found that there is a signature of adaptive evolution on the protein coding sequence of ADH. And all of these lines of evidence have led to it becoming a widely accepted and quite literally a textbook example of adaptation. Precisely stated by Peter Heinstra, he said that a different environmental factor, ethanol, marks the two sibling species. Drosophila melanogaster can be found and breeds in habitats in which fermentations by yeast ADH might generate high concentrations of ethanol. ADH plays the major role in the in vivo elimination of ethanol, and therefore ADH is thought to be a fundamental factor in the difference in habitats between the species. So the classic adaptive hypothesis as it stands seeks to explain variation at these biological levels, and more so it predicts specific causal links. That is how substitutions in ADH affect biochemistry, organismal function, and ultimately fitness in ethanol-rich habitats. However, these causal links have not been tested, and what I'll be talking about is how I tested them using the ancestral sequence reconstruction method. So just to give a brief overview, if we think that adaptive evolution occurred on this lineage, then the two things we need to compare are the adapted protein that exists in Drosophila melanogaster to its ancestral variant prior to the hypothesized adaptation. So the ancestral protein, of course, is ancestral because it doesn't exist, but we can recreate it and characterize it just like we could with a present-day protein. So the technique we use is ASR, and basically we take sequences from present-day Drosophila species. We use a maximum likelihood method to infer a phylogeny, and then we can infer the sequence that corresponds to our node of interest, which is here. So using that, we can recreate and basically test that. Before going on, I'd just like to point out that when I say the Drosophila melanogaster ADH, I'm talking about the ancestral Drosophila melanogaster ADH protein, which corresponds in sequence to the slow allele that many people know about. The fast allele is a more derived form, and I'll also be characterizing that as a positive control because it is known to have higher activity. So basically, we're testing what happened in this evolutionary lineage between the blue and the red dots. And just to show you, we can reconstruct the ADH protein with really high confidence. There's only one ambiguously constructed site, and the alternate state makes it identical in sequence to the simulans protein. And this site just toggles between two states across the Drosophila phylogeny. So the first part of this was testing how the changes in genotype affected protein chemistry. And we did this using a standard ADH assay, where we look at the rate of reaction. And we did this with controlled amounts of enzyme across ethanol concentrations, so we could generate Michaelis-Menten curves and estimate various enzyme parameters. Our prediction is from the classic hypothesis that the melanogaster ADH would have higher activity. However, when we do the experiment, we see that it is the melanogaster ADH in red is statistically indistinguishable from both the ancestral allele in blue and from the simulans allele in orange. The fast allele does have higher activity, which shows that we can detect a difference. And this, we can just look at it at a more fine level and see that this holds per unit enzyme. The catalytic turnover rate is indistinguishable between historical and the melanogaster variants, but the fast is different. And same with the Michaelis constant, which describes enzyme behavior at subsaturating concentrations of ethanol. So at the protein biochemistry level, there does not appear to be divergence in function among on the melanogaster lineage for ADH. And this is not unique to ethanol. I've characterized this across other alcohols. And here, for example, in isopropanol, we see the same pattern qualitatively, although quantitatively it is different for both the catalytic rate and for the Michaelis constant. Okay, so the first prediction of this hypothesis does not hold. However, it is possible that the sequence differences lead to functional differences in the organismal context. For example, a post-translational modification could affect the activity of the protein in the cell. To test this, we, and I use the role of we because it's actually David Lowen, engineered flies where he tested all of the alleles that we tested from purified protein. So these strains differ only in the identity of their amino acid coding sequence. And basically, he looked to see if there was difference in ADH activity if we characterized them from cell extract. And after controlling for protein levels, we see that the maximum velocity, again, is no difference between the ancestral variants and the extant melanogaster variants. Again, we see that the fast protein is different. Okay, so that prediction of the classic adaptive hypothesis also does not hold. But ultimately, the most interesting part is, do these changes affect the organismal phenotype? That is, do the substitutions in the melanogaster ADH confer an ability to tolerate higher levels of ethanol in animals? And to do this, I went to Nebraska and worked with my undergrad mentor, Christy Montooth, under the tutelage of Catherine O'Brien, thank you. And we did some painful experiments where we actually went and figured out what the ethanol tolerance of these transgenic animals were. So first, at the larval life stage, we did this by taking pools of 25 second instar to third instar transition larvae, placing them in vials containing various concentrations of ethanol, and basically measuring how many of them survived eclosion, how many of them lived. So when we take that data and we plot it, we basically get a dose response curve. And from this curve, we can estimate what the ethanol lethal dose is and whether that varies between the different transgenic strains. As you can see here, it doesn't. The difference you would expect is for the red curve, which corresponds to the melanogaster ADH, to be right shifted. It is not. It is, again, statistically indistinguishable from strains carrying the other variants. And this and all of the other data have been replicated with multiple strains, and that is not the main effect. What is interesting here, though, is that the fast allele, a single amino acid difference that distinguishes the fast allele, is sufficient to increase the LD50 at the larval life stage. Qualitatively, we see the same thing at the adult life stage for LD50, although here we can't quite say if the fast allele is more ethanol tolerant or not. But so, at that level, the predictions of the classic hypothesis doesn't hold either. So this leaves us with one other thing. That is, so what was the signature of positive selection then corresponding to? And so the first thing we thought was perhaps it was insufficient sampling in history. For example, McDonald and Kreitman could not characterize polymorphisms to nearly the same extent that we can today. So to see if that was the case, we looked at more polymorphism data. So we added polymorphism data provided to us by Daniel Matute for Drosophila yacuba, which was wonderful, and also by the DPGP2 project. And we see that the signature of selection on the protein coding sequence is still there. However, if we break this down with phylogenetic structure, we see that the signature of selection does not come from where we expected it. That is, most of the non-synonymous replacements come from the Drosophila jacoba lineage, which is difficult to interpret because Drosophila jacoba does not have a remarkably high ethanol tolerance. It behaves pretty similar to simulins and other species in the melanogaster group. You can see that, sorry if the red-green thing is throwing off colorblind people, but basically melanogaster has high ethanol tolerance, high LD50s, and jacoba and simulins have comparable low LD50s. By contrast, on the melanogaster lineage, there's been only one non-synonymous substitution, which is highlighted here on the protein structure. Of course, I think it is worth noting that even though it's only one substitution, we can't rule out the fact that it might have been adaptive, and that is why we did the experiments, because the fast allele, which is also different by one mutation, can affect both protein activity and survival on ethanol. Ultimately, though, we can take all of these lines of evidence and revisit the classic adaptive hypothesis. The classic adaptive hypothesis did what most hypotheses are supposed to do. That is, it specified testable links between phenomena at different biological levels. However, the classic hypothesis was accepted on the basis of correlated lines of evidence that were all ambiguous. And even though they fit into a really intuitive narrative, they were never tested mechanistically. Using ancestral sequence reconstruction and characterization of proteins and transgenic animals, we were able to test the causal links, and we find evidence for none of them. Largely, this raises the point of making inferences about specific adaptive phenomena as it relates to proteins. Because generally, what does happen is if we see multiple lines of evidence, we infer adaptation. Here, we see how intuition can betray us. However, I think one of the cool things is using new technologies like ancestral reconstruction and using them creatively with phenotypic assays that have been done in the field for a long time, we can more directly test specific hypotheses of molecular adaptation. And this seems like something that can be broadly applied to hypotheses of protein adaptation. With that, I'd like to thank the wonderful people I got to collaborate with on this project. That is Christy Montooth, David, and Joe Thornton, and I'm in the Thornton lab, and I get to work with these great scientists every day, as well as the evolution community at University of Chicago. Thank you.